Assalamu alaikum everyone. Are we audible enough? Assalamu alaikum everyone. Are we audible enough? Assalamu alaikum everyone. Are we audible enough? For everyone? Let's move toward the class. Before that, just let us know if you are new at our platform or you have any kind of previous experience. We are starting in 2 minutes. Besides this, the attendance form will be shared in the live chat. Do fill out the attendance form for the certificate. So that by the end it doesn't mess up. Okay, that's good. If you had done one docking project, that meant you have all of the knowledge with the practical skills. This workshop is basically for the undergraduate candidate who doesn't know anything about the molecular docking before moving toward the practical sessions. So let's start our workshop and let's confirm that the screen stays visible or not. Like, is it still visible or not? So that we can move toward the session as it already passed 15 minutes. Is the screen still visible? Okay, let's move toward the session. Drop your questions over to the chat box and we will answer them by the end of the session. For now, focus on the session. Sorry. So here is the Biocode Innovators presenting us a workshop over to the molecular docking, the introduction of molecular docking. Basically, this workshop is arranged for the undergraduates or the candidates of the bioinformatics or biotechnology who wants to get the grip of theoretical concept about the molecular docking and doesn't know anything about the practical one working. About the Biocode Innovators, I hope so many of you are still are already the part of the community, so I don't have to describe longly. I'll give you a brief overview about my community and then we will be moving toward the workshop. Okay. So Biocode Innovators, we are the student-led initiative and we are offering multiple wings for the education purpose or education content with the multiple wings related to the bioinformatics, biotech, AI and data science along with the related fields like microbiology and all of this. So through the workshops or boot camps, there are many more things ahead. We had planned many more things to make your break predictable. As for now, we are offering multiple type of workshops, bootcamp, and internship program, and the research focused program for the training programs for your learning. And in this way, we are bridging up the gap between the biology and the tech. And I am the founder of this Biocode Innovators. My name is Abir Iftikhar, and I am the silver medalist. Okay, screen is changing, right? Now let's move toward. So, as I think many of you doesn't know anything about the molecular docking, as this workshop will be all about the theoretical concepts behind this phenomena, as this is one of the major wing of the bioinformatics. That molecular docking is a kind of important one edge that you can go for the specialization or the master in this wing for the studies. So here we are going to cover up the foundational concepts that will cover up the molecular docking, including its how and which type of molecules we use for the docking purpose, and what are the docking types, along with the workflow, what workflow we follow up, and the applications along with the limitations. So one more time I am explaining it that this is a theoretical workshop. You don't have to install any software, just will give you the concept. So these are the contents. These are the contents of your workshop. That is molecular docking. What are the core concepts, and why do we use the docking for the drug designing? Like, what is the logic behind it, and what are the key terminologies that we use for the docking purpose, and what type of molecules we use for the docking purpose? We are going to check for them, and what are the binding sites, and how do we describe the scores, along with what type of model does it follow up with the interaction whenever the protein and the ligand interact with each other. And there are two types of docking. We are going to discuss it, and along with the docking step, just a guide, and along with the limitations. So, how do we discover the drugs before moving toward this bioinformatics or computational edge? Like, let's say, let's go back toward the traditional times when the drugs take almost 10 to 15 years, or 8 to 10 years, or it cost millions or billions, we can say even trillions if the drug is costly and the experimental cost is too much. So at that time, it according to the traditional way, it take multiple years, along with the too much cost. And as for the too much cost, along with the multiple trial and fail error based work, it doesn't settle well. As for we are living in the advanced environment, and the more advanced environment, the more advanced diseases we are encountering with. So whenever there is any kind of pandemic attack or any kind of severe attack of any of the disease, we don't have the time of 5 to 10 years, or 5 to 7 year, or even 2 to 3 years to cover up or prepare a drug manually or traditionally into the laboratories directly. And that's why we had a move toward this computational method. And this computational method, computational biology method, or we can say the bioinformatics, have gained a spotlight after the COVID. As for the COVID vaccine, was majorly developed using this edge, as if I don't know if anybody knows that COVID vaccine was developed using this molecular docking. As for the docking purpose, we can say that we can design the drug on the computational tools or their sources, and we can even predict if the drug have any kind of severe effect on any kind of metabolic activity of our body, of human body, rather than moving toward the manual designing of drug and then experimentally applying it to the multiple organisms like the rats and the rabbits before moving toward the humans. So in this way, with the computational biology, this has make this edge easier, as we can easily move or predict the drugs or their even their effect or toxicity test on the human body. And if the results finalized, and those results are passed on to the wet lab scientist, and they can easily develop the vaccine or any kind of drug in meantime, rather than going for that 10 to 7 years or billion cost of about repeat or fail area. So in this way, we can easily identify or promise any kind of drug candidates before stepping into the lab or the wet lab, we can say. This will also save our time, our money, our lives, and the also moves us toward the more advancement. Like for the traditional pathway, if we say, let's say we synthesize any kind of drug or the vaccine or any kind of thing like that, and after synthesizing it, we have to move toward the trial method or the test method, we can say. There are multiple species that are considered step by step for the test and trial method, starting from rats to the monkeys. There are multiple candidates or multiple species within this gap line, and we have to make sure that every organism survive with that particular drug or the vaccine we had developed. So this take a lot of time as for the fail and repeat error, and we have to dispose of our discoveries are working again and again, and we have to re-attempt it to synthesize a proper drug in a traditional way. And with a computational way, we can use for the prediction methods or computational methods or the tools to predict the results or predict the drug, its structure, and even its effect over to that particular part of or the particular organ or organ system of the humans we are targeting it. And we can even check for their effect if the drug is if the drug is likely affected by body or any kind of severe allergic reactions. Or in this way, we are also preparing up the personalized medicine, and that is another one important edge. So with the computational path, we can easily simulate or predict or prioritize, or after the validation of our computational drug, we can move that manual toward the wet lab scientist. They can directly develop the drug, and with just one trial and error method, they can simply drop it into the market. So what is basically molecular docking? Molecular docking, we can say that it is a technique, important one technique through which we can predict how the small molecules, like it can be a protein and any kind of ligand. There are multiple type of dockings, like either we are going to dock protein and ligand, and either we are going to dock for the protein and protein. There are different types of wings like this, but molecular docking will show us how we can predict or the small molecules like ligand to any kind of target molecule. That can be any protein. We can also say that protein as a receptor protein. or the receptor target, and like finding its way that where that protein binds on the ligand, or where that ligand binds on the protein. Like we are actually finding out the proper binding sites on which it will give us the good results. Like protein itself offer many of the binding site. Like protein is also the enzyme, the concept we have read in the intermediate, like that the protein are also enzymes, or these um, enzymes do have the particular, you can say, what do we say that, particular binding sites, and those binding sites are easily fitted by the particular type of structures. We can say that ligand for this one. So we are going to cover up that models afterwards, and we have to predict either the results or the drug we are preparing, either it binds directly to the ligand or to its target. What are the different scores and the binding energies? The whatever score shows us, and we have to figure out that which one is the best one candidate, and we can move forward for the manual designing. And besides this, this is one of the most important thing, that these molecular docking techniques have been used in the COVID vaccines and research cancer vaccines and HIV vaccines. These are the key players, and we can say that the core concepts. Like if we are going to talk for the protein to ligand, we have to figure it out that what is protein. We can say that any kind of large biomolecule or biological molecule, we have to pick up the 3D structure of protein that we can easily get from the PDB interface, PDB database, and that protein can be any kind of enzyme, or usually we say it as a receptor that we have to aim it directly, so that it direct with our ligand. Along this, the ligand, we can say a small molecule, chemical structure, or the chemical structural molecule through which we have to bind the protein active site. Okay, so we can say that protein is a big one ball, and that ball has a hole in a moon shape, and in that moon shape the ligand is going to fit. So these the interaction or the ball, the example I gave, the whole structure will be considered with the binding interaction, like how we will check out if there is any kind of hydrogen bonding, the hydrophobic interaction or the interaction allostatic interaction along this to place hold the ligand in a place so it doesn't fade away from the protein. And besides this, how do protein and ligands interact? We had covered these things or these concepts up when we were back in the intermediate, and I think everyone knows about the key model, key and lock and key model concept. Like if there is any kind of protein, we can say that this one is considered as the protein, and this one is the ligand, and we are offering the ligand to bind it directly toward the protein, the active site, and it directly binds toward the active site as if this one is the key, the protein is the key, and the ligand is the protein is the lock, and the ligand is considered as the key to fit in directly. And this whole interaction or interacted binding sites, this one is considered as the binding site, and this binding site had fitted the protein and ligand directly for the interaction. So this whole protein ligand interaction is used further for the docking results. Besides this, there we can say that in the lock and key model there is no flexibility, like we can't change any kind of conformation, as it doesn't offer any kind of allostatic sites, the sites that are other than active sites. And so we can say that these model, or the lock and key model based interaction of protein and ligand, are simple models, or the less realistic. But besides this, let's say for the induced fit model, like the shape will be changed according to the shape of the ligand. Like this is the protein, and it is changing its shape to fit on the ligand on its place. And in this way, we can say that the protein flexes to accommodate, or fixes itself to accommodate the ligand, and this one is considered as the more realistic and the dynamic binding. Along these things, these are the two basic models that assumes are as the rigid one, or the perfectly fitted protein ligand interaction. So we can say that induced fit model is the more realistic one, and as compared to the lock and key model. So, and with this we can say that the binding is influenced by its any kind of shape or the charge and the ligand offers. Does it align with any kind of hydrophobic interaction or hydrogen bonds along this? These are the types of molecular docking. There are two types of molecular docking: a rigid docking or either a flexible docking. If we are having both protein and ligand structures are as fixed structures, we can say that this one rigid docking is for the induced fit model, that the protein and the ligand holds on to its fixed structure and then bind together for the interaction. But this one is we can say that this one is the fast and less accurate for the flexible binding. If there are multiple type of ways, sometimes we don't dock one protein and the one ligand. Sometimes we move toward the multiple docking purpose, like we are docking one protein with the multiple ligands. So in that way, rigid docking doesn't work, the flexible docking works. And the flexible docking, we can say it fits into the induced fit model, and in which we can change up the shape or any kind of simulations and confirmation for the more realistic or computational intensive designs of the drug. So I think the concept is clear. In the rigid docking, we can only dock for the protein or the ligand that stays with its fixed shape and doesn't go for any kind of changing, or we can say the reformation before moving or fixing into each other. And with the flexible one, we can say it is lower. It can adopt any kind of structure according to the ligand shape, or a protein can change its structure, and we can say that it is for the higher accuracy one. I hope till here everything is understood. Just drop out your question into the chat box. We will answer them by the end of the session. Along this, this is a kind of molecular docking workflow. Like you have to select on the ligand. Firstly, you have to prepare up your manual or about what type of project you are going to cover up. It doesn't matter, you will be writing up as a scientific purpose in a first way. Like if you are a beginner in a molecular docking project or molecular docking, do prepare multiple type of small projects for your practice purpose. Don't take your mind like I have to publish it right away. you can't do that. It takes a lot of time, it takes a lot of information gathering along with the citation and references. That is too much for the first attempt. But if you are attempting it at the first time and you want to do it perfectly, go for the small projects that offer you the whole workflow line, and you can save up your work, and afterwards, whenever you get the good grip, you can move toward your research writing purpose. So in that way, you'd have to move toward prepare up your manual. We're going to follow like what are you going to discuss or predict about the disease. Either you are going for any kind of microbial infection treatment, or either you are going for any kind of the tumor treatment. So in this way, you have to select for the ligands, and you have to download the ligand, prepare up your drug candidate. Either it is a multiple ligand you are going to do, or either you are going to do for the protein with a single one ligand. You have to select for your target protein. You have to download it. Besides this, you have to clean up your whole protein and the ligand, and then you have to move toward the docking tools for this purpose, and afterwards docking simulation, and then admit results about the drug likeness to check for the scores and result interpretations. And on this, when we can say that each step is important, you have to carefully analyze all of the results. So the next thing is how do you read your docking results? Like either the first thing, whenever you are going to dock, many of the candidates go for the multiple ligand docking with the single protein. It goes well with the binding affinities. So we can check which one ligand is going well or binding well with the protein. We can pick them up for the further studies. So in this way, we can say that we can check how strong the ligand is binding, depending on its binding energy. Either it is reported one in that I don't have screenshot or image or image, and that way I can show you how do you read it. But let's keep it aside. We can say that the more negative value of the binding energies, we can say that that is the strongest binding. We can pick that up. We can pick that candidate up for the further analysis or the further downstreaming. And then there is a kind of docking score through which we can rank our or check for the multiple ligands. Besides this, it also offers the binding poses. Like there are multiple type of binding poses on a single protein, whether your ligand is binding on the good side or not. This is one of the important factors to check. So we can say that where does the docking makes an impact? We can screen thousands or multiple or hundreds of molecules or compounds to find out the disease head target in meantime. Like it takes time, but it doesn't take years. It takes almost 4 to 5 hours if you are dealing with the thousands or hundreds of candidates at one hit. Along this, you can go for any kind of drug repurposing, like you can find out the new uses of existing drugs, like just like the infections or any kind of diseases are modifying, or mutated one diseases are coming back. The same way the drugs are already existed in the market. So we just have to repurpose that drug so that we can move toward the better option. And in that way, it makes the drug drop into the market in a faster or the cheaper way. Besides this, we can also go for any kind of disease search, like we are having the molecular or all of the genetic information or the data of any kind of diseases, or about any kind of mutated genes or the normal gene, we can compare them up, and we can go for the prediction of any kind of disease, drug, or either we are predicting any kind of models. Besides this is one of the most important thing, that we are moving toward the personalized medicine, that we design on the base of individual generic data or profiles. Mm, these are the limitations, like there are multiple type of scoring functions that are approximate. If your results are more in approximate category, you can consider that it is not the perfect predictors. And protein flexibility, sometimes it is hard to handle if you are using multiple ligands with one protein. Along this, the experimental validation of your drug before signing it as the best candidate is really important. And one more thing is that if the 3D structure of your protein doesn't exist, you can predict it using many of the AI tools or the softwares. Just like AlphaFold offers you the predicted 3D structure of protein if the PDB doesn't offer you the actual structure, so we can develop it through the predicted one week. And with this one, we can also go for the virtual screening in a massive way using the cloud computing one. And besides this, whenever you go for the docking, after the docking scoring results, you have to move toward the simulation and the admit results to check for the more accuracy. So in short, we can say that this is a powerful start or a powerful combination through which we can prepare any kind of drug in meantime with the best outcomes. So here is the conclusion. I hope so everything is understood. As the molecular docking through which we can predict any kind of protein with the protein interaction and any kind of protein with the ligand interaction. Like we are going to check for the molecule interaction with a biological target to check for its ability or effect in the body before moving toward the manual designing. So by understanding all of these protein ligand interactions and ligand interaction and these principles of docking with the scoring results and the admit results and MD simulation results, we can check for, or we can move toward the better therapeutic development results. So I hope so this workshop will have provided you all of the foundational core concepts about the workflows or applications or any kind of limitation, and the workflow line through which you can develop up the molecular docking or project or the computational drug design. Thank you so much. Any kind of question, kindly drop your question into the chat box. We will answer them. And do fill out your attendance form for the certificate. Here is what Biocode Innovators is offering. As this one was a theoretical workshop about the molecular docking, we are offering up the program of 8 weeks about the bioinformatics and the molecular docking, but this one is considered a paid program, as for the instructor is going to put a lot of effort in delivering on each and every session, as every session held on the practical working with about 1 or 1.5 hours. That is too lengthy. Along with all of these descriptions have been written here, that what type of bioinformatic databases or tools we are going to cover, along with your molecular docking, a wing that that we are going to cover in this internship program. This will be starting from July 1, and soon. I think we had already announced the details about this program. Feel free to approach for the details. And in this wing, what you will get, you will have your own GitHub repository. We will guide you about the GitHub account, and you will have your own GitHub repository work, all of yours, submitted over to the structured or clean GitHub repository. You can share with any of your recruiters for the further work, or you can even submit it as the internship um, experience in your university too. Along this, you will get the certificate of completion and the experience letter if you submit your GitHub repository back on the assigned date. Thank you so much. Any question, anyone? Any question, anyone, related to anything at all? I think the link is shared with I'm sharing up the link. Here we have shared up the link of the form. Please let us know if the link works. Do fill out your attendance, and thank you so much everyone for joining today. There are multiple type of tools we use in the docking. We have to move toward the either you want to dock with the Autodock Vina or either you want to dock with the CD dock. There are multiple type of software we use in docking. It depends either you are going for the flexible docking or either you are going for the blind docking. Is the link working or not? You can fill out your form. We are signing off in a few minutes. Thank you so much everyone for joining. Do share the recording link with all of the other candidates who are interested. I use many tools for the docking. I do use the Autodock Vina and I do use the CD dock and along with the MD simulation and admitter. Is everyone filling out the attendance form or not? That's all for today. If anybody still have any questions, we can move toward the group chat. We are signing off from the live chat. No, the set tools aren't paid. The tools I had explained are not paid. You can easily download them in your laptop or in your system. And those are the softwares you have to download. And many of them are also the live or online interfaces or databases through which you can go for the analysis of yours in a direct way. You don't have to download. Anything else, anyone? Anyone else have any question? You can easily install or get these tools from the Google. You don't have to go for any kind of other way around. You can simply write up the name and download all of these tools from the Google. Thank you so much, everyone. Yes, this session is available offline too. You can re-watch it as it was a YouTube live. Thank you so much, everyone.